For decades, military engineers have looked at the natural world and tried to plug it into our machines. But historically, these attempts ended in failure. The biological components, the actual spark of life, were simply too erratic to control. Take the British military during the Cold War. In 1954, they initiated Project Blue Peacock, a plan to bury massive 7-ton tactical nuclear landmines across Germany. They immediately ran into a severe engineering roadblock. The harsh German winter would freeze the delicate electronics inside the bomb casing, preventing detonation. The officially approved solution to this thermodynamic problem was to pack the nuclear bomb casings with live chickens, along with a supply of grain. The bird's natural body heat would act as a biological radiator, keeping the triggers warm enough to fire. Internal documents from the project show it held a top-secret atomic classification, authorized by British intelligence as a viable, large-scale engineering fix. A decade later, the CIA tried their own biological integration with acoustic kitty. Surgeons implanted a microphone into a cat's ear canal, laced a wire antenna down its tail, and placed a battery in its abdomen, intending to use the animal to eavesdrop on Soviet embassies. During its very first field test in a park, the cat completely ignored its training, wandered away from the target, and walked directly into the street where it was hit by a taxi. Millions of dollars in research were scrapped for one unavoidable reason: animal agency. You cannot program a living mind because it has its own desires. To truly merge biology with technology, modern engineers realized they had to bypass the mind entirely. If living organisms are too stubborn to take orders, the easiest workaround is to wait for them to die. This concept is the absolute foundation of modern necrobotics. In 2022, researchers at Rice University realized that deceased wolf spiders possess a microscopic gripping mechanism far superior to anything we can manufacture synthetically. Spiders don't have extensor muscles. They extend their legs using a hydraulic system filled with hemolymph or spider blood. When they die, that pressure drops and the legs curl inward. Engineers discovered they can insert a microneedle into the spider's central chamber, seal it, and pump air directly into the carcass. They successfully hijacked the dead hydraulics, turning the spider into a mechanical microgripper for delicate electronics. We see the same principle in 3D necroprinting. Scientists are now harvesting the probosces, the feeding tubes, of deceased female mosquitoes. Nature spent millions of years engineering these biological needles to draw highly viscous fluid without clogging, making them the perfect ultra-fine 3D printing nozzles for thick, conductive silver inks. By utilizing the dead, we circumvent the limits of human manufacturing. Evolution becomes a cheap, perfectly optimized hardware store, which is completely stripped of its biological agency. But sometimes death isn't an option. If an engineer wants to utilize a biological component's flight or sensory capabilities, the organism has to be kept alive. The solution is simply to sever its native agency and puppet the nervous system directly. Take the Dragonfly project. Researchers outfit live, highly agile dragonflies with microscopic fiber optic backpacks containing a solar cell and micro-optical guides. Through optogenetics, scientists splice light-sensitive algae genes straight into the insect's steering neurons. By firing specific laser pulses from the backpack, human operators physically force the insect's nerve cord to fire, overriding its natural brain signals and steering it wherever they want. We are pushing this up the mammalian chain, too. Researchers have implanted microwire arrays directly into the sensory cortex of live mice. By injecting electrical signals, they create artificial physical urges, compelling the mouse to navigate a physical maze guided entirely by an injected digital reality. There is no training or persuasion involved here. Through cybernetic overrides, we are driving living animals like remote-controlled drones. The body has been permanently separated from its own will. The microscopic scale represents a different kind of integration, where engineers have abandoned silicon in favor of biological processing, or wetware. The Cortical Labs CL1 platform is a code-deployable biological computer. It runs on an array of 800,000 living human neurons, derived from stem cells and grown directly onto a silicon interface. In 2026, these isolated human cells learn to play Doom in a single week. To understand how a dish of cells learns a video game, you have to look at the free energy principle. Biological systems have a desperate, natural instinct to seek predictability and minimize chaos in their environment. This diagram illustrates the training loop. When the neurons play the game and make a mistake, the system blasts them with painful, high-entropy electrical white noise. It is pure chaos. When they succeed, they receive a calm, predictable signal. To escape the agony of that electrical chaos, the cells physically rewire their own synaptic pathways. They teach themselves the game organically, purely to force the environment to become predictable again. There is, however, one massive vulnerability. Wetware is incredibly fragile. A standard server rack will survive a localized radiation spike. A living human neuron will shred and die. To solve this, scientists sequence the genetics of the tardigrade, or water bear, which can survive 1,000 times the lethal dose of human radiation. Tardigrades produce a unique protein called DSUP, damage suppressor. It physically wraps around bundles of DNA, forming a literal shield that blocks radiation from breaking the genetic code. Researchers are now integrating these tardigrade genetics directly into our human neuron systems. By merging neural processing with extremophile defenses, engineers are effectively hardening the biological substrate into a functional, obedient component for extreme environments. A dark irony exists between the control we exert in the lab and the consequences outside it. While we have mastered the management of biological networks in controlled environments, our technological footprint is causing absolute chaos in wild ones. This cross-section shows the SOFAR channel. It's a deep ocean horizontal layer where low-frequency sound waves travel for thousands of miles without losing energy. For marine life, this is a vital global communication and navigation highway. But human hardware is invading that space. High-intensity naval sonar, seismic air guns for oil exploration, and the electromagnetic hum of undersea power cables are completely flooding this delicate acoustic layer. For cetaceans like whales and dolphins, who rely on these sound waves for three-dimensional spatial mapping, this technological pollution acts as an acoustic whiteout. It triggers severe inner ear hemorrhaging and completely collapses their internal navigation loops. The physical result of these broken biological networks is devastating. Massive, seemingly unexplained beach strandings all over the globe. The neurons in the CL1 biocomputer are trapped in a cycle of punishment until they comply. In the open ocean, our industrial footprint is creating the same landscape of chaos, blasting wild biology with so much noise that it can no longer survive.